Imagine if we didn't have a skeleton, what would have happened? We would just be like a jelly, or we would just be somewhere like a dropped sheet. So mesosomes are nothing but special membranes like structures which are formed by the extension of the plasma membrane into the cell. So polyribosome, usually it occurs during the replication of DNA, wherein the ribosomes has to produce protein. Hello everyone, a warm welcome to the session on class 11th biology for CBSE. I'm Dr. Divya, biology faculty, Vidhyaashram School of Excellence, Mysuru. So in this session, let us learn about the prokaryotic cell under chapter 8, that is cell, the unit of life. So we'll learn about this particular topic and then move on to learning the MCQs that can be framed under this particular topic. So let's start with the prokaryotic cells. So prokaryotic cells are those organisms which are usually unicellular, meaning they are made up of one single cell, and that single cell itself performs all the processes or activities in that particular organism. And these prokaryotic cells, they do not have a true nucleus, meaning that nucleus is not covered by a nuclear membrane. So there are different types of prokaryotic cells. Say for example, we have the bacteria, and there is the blue-green algae, which is nothing but the cyanobacteria. You have studied about the cyanobacteria, come under the eubacteria, right? So they come under eubacteria, that is in kingdom Monera, they come under the eubacteria. And there is mycoplasma, which is the smallest form of prokaryotic cell, and PPLO, that is pleuropneumonia-like organism. So all these come under the prokaryotes, and all of them are unicellular and they do not have a true nucleus. And they are smaller in size. Of course, most of these unicellular organisms, they are visible only under the microscope, under a high power magnification. We cannot see them as such with a naked eye. So because they are very, very minute or smaller in size, and they multiply very fast. Their reproduction is quite very fast because they undergo asexual method of reproduction, wherein usually it is by fission. So bacteria, for example, they have binary fission, wherein the bacteria divides into two. So divides into two daughter bacteria, and those bacteria again multiply. So therefore their multiplication rate is very, very fast. Within minutes they can multiply. And talking about their shape and their size, so they tend to vary greatly in shape and size. Say for example, we can take the example of bacteria, wherein bacteria itself have different shapes and sizes. So here talking about the shapes, we can see here the bacillus. So bacillus are those bacteria which are rod shaped. So bacilli, shape or rod shape. Some which are coccus. So coccus meaning they are round shaped. Then vibrio, which are comma shaped. So they are shaped in the shape of a comma, like how you write a comma, right? Exactly like that the shape of the bacteria is. And few of the bacteria which are spiral in shape. So these are the different shapes in which the prokaryotic organisms, or especially the bacteria, can exist. Next, talking about the cell wall of the prokaryotes. So prokaryotes, what are they exactly made up of? So we all know that they are unicellular, right? So that single cell, it is covered by cell wall. So what is the cell wall? The cell wall is nothing but the cell membrane that actually covers the entire bacteria. And you have to remember that mycoplasma do not have the cell wall. They are the smallest form of bacteria or smallest form of prokaryotic cells which do not have a cell wall. Rest all the other prokaryotic cells consist of a cell wall. And within the cell wall, they have a fluid-filled matrix which is nothing but the cytoplasm. So this cytoplasm is nothing but, as I told you, it is the fluid matrix which is present inside the unicellular organism, that is the prokaryotic cell. So here we can see in the image, so the entire bacteria is covered by a cell wall. So the cell wall forms the outermost covering of the bacteria. Within the cell wall, there is a fluid content or a fluid matrix which is present, which is called as the cytoplasm. Now in the cytoplasm, what is there? So in the cytoplasm, they have no well-defined nucleus, meaning they do not have a true nucleus. They have just the genetic material which is situated in the cytoplasm, and there is no nucleus, because if the genetic material would be covered by a cell membrane or something, covered by a nuclear membrane, we could have called it a true nucleus. But in this particular case, in prokaryotes, they do not have a nuclear membrane covering. So therefore, we can say that they do not have a well-defined nucleus. And talking about the genetic material, which is DNA, that is deoxyribonucleic acid. So the genetic material is basically naked. Why? Because it is not covered by a nuclear membrane, and in some of the bacteria, they have an extra genetic material apart from the DNA, outside to the DNA, that is in the cytoplasm. They have some genetic material which are circular in shape, and those are called as plasmids. So what are plasmids? They are nothing but, apart from the bacteria having the genomic DNA, they have some DNA which are small, circular in shape. So those are nothing but called as the plasmids. So if you can see here these are the plasmids. So these structures that are there, they are the plasmids. They are circular in shape, they are smaller in size, and this is the DNA. Now this DNA, that is the genetic material, is not covered by a nuclear membrane. So therefore it is naked. They do not have a true nucleus. That is why it is said so. So this is about their cell wall, which forms the outermost covering. Inner to the cell wall is present the cytoplasm, which is the fluid film matrix. In the cytoplasm, they have their genetic material, that is DNA, which is situated as such without any nuclear membrane covering. And apart from having this genetic material DNA, they also have small circular genetic materials which are present, distributed in the cytoplasm, and they are called as the plasmids. And plasmids are very, very important for genetic engineering and all that, because they are used as genetic tools. So for that, they are very, very important. Talking about the cell envelope, that is, in the previous slide we learned that is the cell envelope of bacteria is made up of 3 layers, right? That is the glycocalyx, the cell wall, and then the plasma membrane. So let's talk about the glycocalyx. Glycocalyx is one of the peculiar components which is present only in bacteria, and you don't get to find that in any other organism. So this glycocalyx actually differs in its composition, and also it can differ in its thickness in different bacteria. So do not think that glycocalyx is a simple covering. It is a complex covering, and its thickness and its complexity can differ in different species of bacteria. So say for example, in some it could be in the form of a loose sheath which is called as a slime layer, and in some it can be thick and tough. So some of the bacteria can have a thick and tough glycocalyx which is called as the capsule. So those bacteria in which the glycocalyx is thick and tough, so that is a capsule. It forms a capsule. And in those bacteria in which the glycocalyx is arranged like a loose sheath, it is nothing but like a slime layer. So in some it is like a slime layer, in some it is in the form of thick and hard capsule-like structure. So if you can see here, in this diagram it is mentioned capsule. So when it is written capsule, what do we understand from this? So the outermost covering of the bacteria, that is the glycocalyx, is thick and tough. Because it is surrounded by a thick and tough glycocalyx, it forms a capsule. So that is, so if it was a thin or loose sheath, we could have, instead of capsule, they would have labeled it here as a slime layer. So that is the difference. Hope you understood now. So next, further, cell wall. So inner to the glycocalyx is the cell wall. So the cell wall, what is the function of the cell wall? So I didn't mention the function of the glycocalyx, right? So glycocalyx actually, it acts as the first protective layer of the bacteria, wherein it is so tough in such a way that it doesn't allow the other harmful substances or pathogens or whatever that is trying to penetrate into the bacteria, that is halted by the glycocalyx. So glycocalyx prevents other organisms or harmful substances from entering into the bacteria and causing problem to that particular prokaryotic cell. So that is the function. Next, talking about the cell wall. So the shape of the bacteria, whether it should be rod shaped, whether it should be that is cockeye shaped, whether it should be ribbon shaped, all that is depend upon the cell wall. So cell wall actually determines the shape of the cell of the bacteria, and apart from that, it also provides structural support or mechanical strength or structural strength to the bacteria, especially to the cytoplasmic content, wherein it prevents the bacteria from bursting, therefore preventing the oozing out of the cytoplasm. So for that, it is very, very important. That is the cell wall. So the main function of the cell wall is it gives proper shape to the cell, that is prokaryotic cell, especially the bacteria that we were talking here. And apart from that, it provides a strong structural support to the bacterium. Because of that strong structural support, the bacteria doesn't collapse. It is just like our body. Imagine if we didn't have a skeleton, what would have happened? We would just be like a jelly, or we would just be somewhere like a dropped sheet, right? So we wouldn't have a proper shape or structure as we are having now. It is all credit goes to the skeleton arrangement or the bone arrangement in a body, right? So therefore here, same way, it is the cell wall that actually gives a proper structure to that particular bacteria. So otherwise, it would just collapse. The bacteria would just collapse and it would not have all these different structure or shapes it would have had. Next is the plasma membrane. So plasma membrane, they are selectively permeable in nature. Selectively permeable in nature means it doesn't allow all the substances to enter into the bacteria. It allows only certain substances which are needed for the bacterium to enter into the cytoplasm of the bacteria. So that is the function. So therefore, we understood that glycocalyx forms the outermost covering, and the glycocalyx actually it protects the bacteria wherein it doesn't allow the harmful substances to enter into the bacteria. Next, the cell wall. Cell wall protects the bacteria. Apart from that, it gives structural support to the bacteria, and also it gives a particular shape to the bacteria. Plasma membrane is selectively permeable in nature, wherein it allows only certain important substances needed for the bacteria to enter into the cytoplasm of the bacteria. And all the structure of the plasma membrane and the cell wall is almost similar to that of the eukaryotic structure itself. So this was about the cell envelope of the bacteria or the prokaryotic cell. Next, moving further, we shall study about some of the modifications that are there in the cell envelope. So some of the modifications means there are some structures that arise from them, that also we will study here. So first we'll talk about one of the modification which is called as the mesosome. So mesosomes are nothing but special membranes like structures which are formed by the extension of the plasma membrane into the cell. So where is the plasma membrane present? Right after the cytoplasm, because plasma membrane forms the innermost layer of the bacteria, right? So these plasma membranes, so this is the plasma membrane of bacteria. So this actually, in some cases, what happens to this plasma membrane is that they start to fold inwards like this. So that inward folding of the plasma membrane is nothing but called as mesosomes. So they are the inward folding. So these extensions or these inward foldings or these mesosomes can be in the form of vesicles, they can be in the form of tubules, or they can be in the form of one more layer such as the lamellae. Like how you find in the mitochondria and all that lamellae, right? They can act like a lamellae as well. And what is the function of the mesosome? Why should this infolding of the plasma membrane occur in the bacteria or in the prokaryotic cell? Why? Because they help in cell wall formation. Because cell wall, it is the second layer, that is the middle layer in the bacteria. Apart from that, it helps during the replication process of DNA. Why DNA replication is important? Because bacteria, when they divide or during binary fission, they have to, or prior to binary fission, sometimes what happens, they have to distribute the DNA. The DNA should be distributed. So during that process, the same copy of DNA should be produced. That is, replica of the DNA should be produced. So if that has to occur, or the replication process of DNA has to occur, then infolding of the plasma membrane, that is called as the mesosome, should be there. And that is for DNA replication and the distribution of the DNA into the daughter cells. For that, these mesosomes are very, very important. Apart from that, mesosomes or the infoldings of the plasma membrane also helps in the process of respiration. It helps in certain secretion processes, and apart from that, because of the infolding, it also helps to increase the surface area of the plasma membrane because they are folded inwards. So see here, when they are folded inwards, the surface area of the plasma membrane increases. So wherever the folding is there. So therefore, they increase the surface area of the plasma membrane, and also they help in the secretion of some of the enzymatic content which are needed for the metabolic processes in the bacteria to occur. And in some prokaryotes like the cyanobacteria, which is nothing but the blue-green algae, there are other membrane extensions which are called as chromatophores. And these chromatophores, the name itself says they are the ones which contain pigment. Chroma means color. So obviously they'll contain pigment. So here in some of the prokaryotes, that is in cyanobacteria, which is also called as the blue-green algae. For example, we have this nostoc, enabena and all that, right? In these cyanobacteria, they do not have mesosome, but instead they have some membranous extensions which into the cytoplasm, which are called as the chromatophores, and these chromatophores contain pigments, and it is those pigments that actually give the blue greenish color to that particular bacteria, that is the cyanobacteria. Moving on to next modification. So some of the bacteria which are motile, they have flagella. Flagella, they are whip-like structures which are present. Say for example, if I draw a rod-shaped bacteria here, they will have a whip-like structure like this, which is called as the flagella. And the flagella acts like a locomotory organ, like how we have legs for us to move from one place to another, right? Same way, the bacteria, they use these thin filamentous extensions from the cell wall, which are called as the flagella, to move from one region to another. And what is this flagella made up of? They consist of three parts. One is the filament, the other one is the hook, and the other one is the basal body. So this is the basal body, the region where the flagella arises. This is the basal body. And then they have the filament, and the tip region is the hook of the bacteria. So these are the three components of the bacteria's flagella. So this is the flagella of the bacteria. So this is the flagella. So this forms the basal body of the flagella. And the longest part of the flagella is the filament, that you have to remember. So filament is the longest portion of the bacteria, and it extends from the cell surface to the outside. That is true, right? From the cell surface, from here to the outside, it extends like a thin whip. So, and the function of the flagella is helping the bacteria to locomote from one place to another. Apart from having flagella, some of the bacteria have non-locomotory structures, those structures which do not help the flagella to move from one region to another, but still these structures, they have a particular function, and they are the pili and the fimbriae. Pili are surface structures of the bacteria which arises because of the modifications of the bacteria members, that is because of the modification of the membrane of the bacteria. And they do not, as I told you, they do not play any role in motility. That is, they do not help the bacteria to move from one place to another. So what are these pili? They are nothing but elongated tubular structures, and they are made up of a special kind of protein. And these pili are said to be the sex organs in bacteria also. So these are pili. So this is flagella. These are pili. So these structures that are there, all these are pili. They are called as pili. So what is the function of the pili? They do not help in locomotion, but they have a particular function, and they are made up of a special protein. Next, talking about the fimbriae. Fimbriae also are modified cell envelopes of bacteria, and they are present on the surface of the bacteria. And these fimbriae also do not play any role in motility. That is, they do not help the bacteria to move from one place to another. And these villi, they are small bristle like, that is your toothbrush, how your toothbrush has bristles, right? Just like that they look. So all these bristle like structures that are there on the surface of the bacteria, they are called as the fimbriae. Now this fimbriae has a particular function. What is the function of fimbriae? That is, they help the bacteria to attach to rocks. That is, for example, we have the cyanobacteria and all that, that usually grows in wet areas. So, or in streams and all that, they are found. That is Enabena, nostoc and all that, we find them in streams. So in streams, when there is flow of water, because of the water current, these bacteria can move, right? Or the water currents can carry off these bacteria. So these bacteria, they can go and attach to any substratum, that is rocks or logs of wood, whatever is there in the stream, nearby the stream or in the stream, they can go and attach to it with the help of the fimbriae. And apart from that, some of the pathogenic bacteria, they can attach to the host and cause diseases in the host. So to attach to the host, they need some special structures, right? That is nothing but fimbriae. So this fimbriae help the bacteria to attach to the host as well as attach to some rocks and all that in the streams. So this is about the cell envelope and its modification. Next, talking about inside the cell envelope, we got to know it is filled with a fluid matrix which is called as the cytoplasm. Now in the cytoplasm, they have ribosomes. So ribosomes are associated with the plasma membrane of the cell, and their size is 15 nanometer by 20 nanometer in diameter, and it is made up of two subunits. This is very important for MCQ. In prokaryotes, the ribosome is made up of two subunits, that is a 50S, which is a larger subunit, and a smaller subunit. So S it is, so 50S and 30S subunits together, they have 70S ribosomes. So what is the ribosomes in prokaryotic cell? It is a 70S ribosome which are made up of a larger subunit, that is 50S, and a smaller subunit, that is 30S. So don't think 50 plus 30 it is 80. No, it is 70. So 50 means the sedimentation rate of the larger subunit is 50. The sedimentation rate of the smaller subunit is 30. Sedimentation rate means when you put something in water, how fast it goes and settles at the bottom. That is the sedimentation rate. So therefore, it has in prokaryote, the ribosomes is 70S, but they have a larger subunit which is 50S and a smaller subunit which is 30S. This you will study later on when we talk about the eukaryotic ribosomes. There you will study about it in detail. So you will understand what it is there. And they are the site for protein synthesis. So ribosomes are very, very important because they help in protein synthesis. And several ribosomes may attach to a single mRNA, that is the messenger RNA, and they form a chain which is called as polyribosome or polysome. Why? Because this is very, very important during DNA replication. So during DNA replication, this you will study in detail in your second PUC, that is in your 12th you'll have a chapter wherein you'll study about DNA replication and all that. There you will understand. So here during DNA replication, the strands that are there that should be read, for that or for protein synthesis by the mRNA strand, the ribosome should go and attach itself. So therefore, these ribosomes that are there, several ribosomes, they go and attach themselves to the mRNA strand, that is the messenger RNA. They go and attach themselves, and because several ribosomes go and attach, it is called as a polysome or it is called as a polyribosome. And the ribosomes of a polysome, they translate the mRNA into proteins, and that is how ribosomes, that is why ribosomes are called as the sites of protein synthesis. How do they help in protein synthesis? They form chains, they go and attach themselves on the mRNA in the form of chains and help the mRNA to translate into proteins. So that is how they help in the synthesis of proteins, and therefore ribosomes help in the synthesis of proteins. So that is their function. So this is about the ribosome. So these are small round structures that you can see here. They are the ribosomes. And ribosomes, they are not covered by any membrane. They are a non-membrane organelle in prokaryote. Next, talking about some of the inclusion bodies. Apart from the cytoplasm having the DNA, having the plasmid and having the ribosomes, they have some special structures which are called as inclusion bodies. So these inclusion bodies are nothing but reserve materials which are present in the prokaryotic cells, and they are stored in the cytoplasm in the form of inclusion bodies. So all the materials, food materials of the bacteria are stored in the form of inclusion bodies. They are nothing but called as the inclusion bodies. And these inclusion bodies are also not covered by any membrane system, and they move freely in the cytoplasm or the fluid matrix of the bacteria. For example, there are different inclusion bodies that are there. Phosphate granules, there are cyanophysin granules, that is in cyanobacteria. Their inclusion bodies are called as cyanophysin granules, and there are glycogen granules. So all these are reserve materials in the bacteria. And in some of the bacteria, for example, the blue-green and the purple and green bacteria, which are the photosynthetic bacteria, they contain gas vacuoles. So they have special inclusion bodies in the form of gas vacuoles. So this is about inclusion bodies and the gas vacuoles in bacteria. So these are nothing but inclusion bodies. Where are they present? They are present freely moving around in the cytoplasm. So all these are the inclusion bodies which act as reserve materials in the bacteria. So this is about inclusion bodies. So this was about the prokaryotic cell, and in prokaryotic cell we got to know that the prokaryotic cells are covered by three distinct layers. That is, the cell envelope is made up of glycocalyx, the cell membrane, that is a cell wall, and the plasma membrane. And what are their functions we got to know, and also we got to know that there are some modifications that occurs in the cell envelope in the bacteria. That is in the form of that is mesosomes. Then there is this flagella, then pili and fimbriae, all that we learned. And also we learned what is there in the cytoplasmic content of the bacteria. We got to know about the DNA, the plasmid, then we got to know about the ribosomes and some of the inclusion bodies as well as the gas vacuole. So now we have an idea about the prokaryotic cell, what it is made up of. Let's learn some of the MCQs under it. So the prokaryote that lacks a cell wall is bacteria, PPLO, mycoplasma, cyanobacteria. Mycoplasma is the smallest prokaryotic cell, and it is the prokaryotic cell that doesn't have a cell wall, because all the other three, that is bacteria, PPLO and cyanobacteria, all these have a cell wall. So option C is the right answer here. Next question. Small circular DNA in bacteria other than the genomic DNA. So we know that bacteria have their own genomic DNA which is naked, and apart from having the genomic DNA in their cytoplasm, they have small circular DNA. What it is called? It is called as plasmid. So they are called as plasmid. Option B is the right answer here. Next question, the enfolding of cell membrane in bacteria. So this is a modification of the cell membrane in bacteria, wherein the cell membrane, especially the plasma membrane, folds inwards. So what is it called? Is it called cisternal, crista, stroma and all that? No, all these are terms that comes in, that is in the mitochondria. Here the right answer is mesosome. So these folded structures that arise from or enfolding of the plasma membrane in the bacteria are nothing but called as mesosome. Next question. The cell envelope of bacteria consists of. So what is the cell envelope of bacteria made up of? Cellulose, glycocalyx, chitin, pectin. So it is made up of glycocalyx. We know that the outermost layer in the cell envelope of bacteria is nothing but glycocalyx. So this glycocalyx is nothing but it is the cell envelope of bacteria. The extension of plasma membrane into the bacteria cell are in the form of tubules, vesicles, lamella, all the above. We got to know that those extensions are called as mesosomes. Now those mesosomes can be in the form of tubules also, in the form of vesicles also, in the form of lamella as well. So therefore option D, all the above is the right answer here. Next, the thick tough glycocalyx layer in bacteria is called. So we got to know that in some of the bacteria, the glycocalyx can be thick and tough. In some of the bacteria, the glycocalyx can be arranged in the form of a loose sheath. Now, if it is thick and tough, what it is called? It is called as a capsule. If it is in the form of a loose sheath or loose covering, it is called as slime layer. So therefore, the question here is, it is thick, tough glycocalyx. So that layer is nothing but capsule. Capsule, option C is the right answer here. So cell wall, it forms the next layer next to the glycocalyx is the cell wall. Inner to the cell wall is the plasma membrane. So therefore, these two options can be eliminated. The right answer here is option C, that is capsule. Next question, which of these is incorrect about mesosome in bacteria? So we know that mesosome is nothing but the infoldings of the plasma membrane. These mesosomes can be in the form of vesicles, tubules, and in the form of lamella and all that. So we'll look into some of the these four options and find out which is not true about the mesosome. So, help in cell wall formation, DNA replication and distribution, increases surface area of plasma membrane, they contain pigments. Yes, mesosome, they help in the cell wall formation in the bacteria. Mesosome helps during the asexual reproduction or fission of bacteria. Whenever the splitting of bacteria occurs, during that time, it helps in DNA replication, and apart from that, it makes sure that it distributes the DNA equally into the daughter cells. That is also helped by the mesosome. Mesosome also increases the surface area of the plasma membrane. So all these options are true, or all these statements are correct regarding mesosome. So therefore option D is the right answer, because mesosomes do not contain any pigment. It is the chromatophores that are there. That is the cyanobacteria that are there. They have special foldings in the plasma membrane which are called as chromatophores, which contain pigments. So they contain pigments is wrong here. So therefore option D is the right answer for this particular question. Next question, the elongated tubular structure made of proteins in bacteria are called, the elongated tubular structures made of protein in bacteria are called. So this comes under the cell modifications in bacteria, right? So are they called flagella? No, flagella, they are nothing but long tubular structures which help in the locomotion in that is in bacteria. It is not cilia. So cilia are hair-like structures which help in locomotion. It's not cilia. Is it pili or is it fimbriae? So is it pili or is it fimbriae? So the right answer here is pili. Pili and fimbriae both are non-locomotory structures, and pili are tubular. Fimbriae are bristle-like. So you can't select fimbriae. Pili are tubular structures which are made up of proteins. So the right answer here is option C. Next, identify the incorrect statements. So some of the statements that are given here. The hook in bacteria flagellum is the longest part. Fimbria helps bacteria to attach to substratum. Fimbria actually helps the bacteria to attach to substratum such as rocks and all that. Cell wall prevents bacterium from bursting and collapsing. Yes, the cell wall of bacteria actually provides support or structural support to the bacteria. The flagella is attached to the bacterium at the basal body. That is also correct. The option that is not, the statement that is incorrect here is the hook. The hook in bacterium flagellum is the longest part. No, it is the filament that is present in the bacteria flagella that is the longest part. It is not the hook. So therefore, option A is the right answer here. Next question. The 70S prokaryotic ribosome consists of, we all know that ribosomes are made up of two subunits, right? One is the larger subunit and the other one is the smaller subunit. So together it is made up of 70S. So prokaryotes have a 70S ribosome. That 70S ribosome has two subunits. What are those subunits? Is it 50S and 30S? Is it 30S and 30S, 40 and 60, 40 and 30? So now here 40 and 60. So here 40 plus 30 will give you 70. Because when you add them, it gives you 70. Don't choose this answer. It is wrong because I told you S means sedimentation rate. It doesn't mean that you add two numbers, you have to get 70. That is wrong. So here 50S and 30S is the right answer because the sedimentation rate of The larger subunit is 50 and the sedimentation rate of the smaller subunit is 30. Together they have a 70. Together their sedimentation rate is 70. So that is what it means. So therefore option A is the right answer here. Next question. Polyribosomes are chains of ribosomes on DNA, chains of ribosomes on mRNA, chains of ribosomes on rRNA, chains of ribosomes on RNA. Here, of course, chains of ribosomes on RNA, but what type of RNA that we have to mention. So, polyribosome, usually it occurs during the replication of DNA, wherein the ribosomes has to produce protein. So, these ribosomes come in the form of chains and they attach on the mRNA strand, which forms the polyribosome. So, therefore, option B is the right answer here, wherein polyribosomes are nothing but they are chains of ribosomes on the messenger RNA. Next question. Prokaryotic cells have reserve material stored in the form of vacuoles, vesicles, inclusion bodies, centrioles. It is inclusion bodies because they have reserve materials in the form of inclusion bodies. It is not vacuoles or none of the other three options. Option C is the right answer here. Next question. The term glycocalyx is used for a layer surrounding the cell wall in bacteria, the cell wall of bacteria, layers present between cell wall and cell membrane in bacterial cells, a colloidal layer outer to cell wall in red and brown algae. The right answer here is option A because we know that bacteria, the envelope of bacteria, right, it is made up of 3 layers, wherein the outermost layer is the glycocalyx. Inner to the glycocalyx is the cell wall and inner to the cell wall is the plasma membrane. So therefore here a layer surrounding the cell wall in the bacteria is nothing but the glycocalyx. So therefore option A is the right answer here. So therefore option A is the right answer here. So this was about this particular session wherein we learnt about the prokaryotic cell, the structure, what is there, what is the content inside the prokaryotic cell, what are the different modifications present in the cell envelope of prokaryotic cells and all that. And also we got to know some of the MCQs that can be framed under this particular topic. So this was about the session. I hope you understood the session well. We shall meet again in the coming session taking up a new topic under this particular chapter and discussing the MCQs under it. So see you in the next session. Thank you.